Welcome to the Brain and Behavior Research Foundation monthly Meet the Scientist webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. B.J. Casey will present a webinar entitled Early Life Stress, Long-Lasting Impact on Brain Circuitry and Behavior. The Brain and Behavior Research Foundation is dedicated to funding research around the world to identify the causes, improve treatments, and ultimately develop cures and preventative techniques for mental illness. The Foundation has awarded more than $320 million in research grants over the last 25 years. I'm delighted to introduce Dr. B.J. Casey. Dr. Casey is Director and Sackler Professor of Developmental Psychobiology for the Sackler Institute for Developmental Psychobiology at the Weill Cornell Medical College. Dr. Casey has been a pioneer in novel uses of brain imaging to understand childhood disorders, using imaging to develop fundamental models of typical and atypical brain development. Her research focuses on the development of the brain circuitry involved in learning and behavior regulation and how disruptions in these brain systems can give rise to a number of disorders in children. Her recent discoveries of the biological factors related to vulnerability and resilience to mental illness during the early postnatal and adolescent periods have important implications for the timing and development of new interventions and treatments. Dr. Casey is a member of our Foundation's Scientific Council. The Council identifies the most promising ideas to fund with research grants each year. Today's webinar will be an interactive event. We'll start with Dr. Casey's presentation, followed by a question and answer period. To submit your questions, please use the Questions tab of the webinar control panel. You can submit your questions throughout the presentation. As your moderator, I will present your questions to Dr. Casey and will address as many as possible. And now, I'm pleased to present Dr. B.J. Casey. Okay.
Thank you, Jeff. Let me just pull my slides up quickly. And I just want to say, as a member of the Scientific Council and as someone who sits on many advisory boards for many foundations, this is truly one that is a partnership among basic scientists, clinicians, and also families with a real effort and a real sincere effort to promote mental health and to also work together to ameliorate mental illness, not simply in terms of thinking about treating these disorders, but really beginning to try to prevent many of these symptoms. And the only way we can do that is to identify risk factors which put an individual at greater risk. And so today I wanted to focus on research from our laboratory and highlight the work of others that has really been seminal in identifying particular factors that might put an individual at greater risk, and to help us drive our research into new ways to think about better ways of treating these individuals and intervening. So thanks to the Foundation, and also thanks to each one of you for being on this webinar today. So let's just begin by highlighting why it's so important to identify risk factors, particularly in young people today. And basically, that is in large part, I wanted to highlight. Let's just see if I can get the next slide up.
Just click one more time. Exactly. Perfect.
So basically, mental illnesses impact as many as 1 in 5 young people today, and they peak during the adolescent period. Each time I present these statistics, there is a visceral response that I have. As the mother of a teenage son, he is 19, has had many challenges in trying to meet all the social demands, and also just to negotiate new adult roles as he's transitioned by being predominantly in his parents' care to now trying to be independent and on his own care. We know that when individuals have difficulties with this, and that can lead to a heightened risk for mental illness, that if these problems are not addressed, if they're not treated, that these symptoms and disorders can go on to lead to not only mental illness, but also physical illness. We also know during this time, one of the number one causes of death, or leading causes, is that of suicide. And so we're always trying to think of ways to enhance our research to identify those individuals at risk. But I just want to show you a simple cartoon that highlights for you how many mental illnesses are either peaking or are actually emerging during this adolescent period, right after about 10 to 20 years of age. And if you look for any mental illness, the average age of onset is right around this adolescent period of time. Today I thought I'd talk about risk factors for the most common. Those involve anxiety and mood disorders, and also any of the disorders that are related to stress at some level. Because these two have a high prevalence of 1 in 10. And while we have a number of medications that we can use, if we turn to also behavioral therapies, such as exposure-based cognitive behavioral therapies and other behavioral therapies, we're only seeing about a 60% improvement in symptoms in some of these individuals. And let me just highlight again, if these individuals aren't treated in a way that's successful for them, this is going to lead to longer-term symptoms, as I mentioned before, that can be chronic and also lead to physical illness. So really the objectives and what I wanted to describe today is to unpack for you what are the typical changes that are occurring in brain and behavior at this time when we're seeing such a peak in the emergence of mental illness, and then also to describe for you studies that suggest risk factors for how an individual during this adolescent or developmental transitional period can become at greater risk for mental illness. This next slide is a simplified cartoon of all of the different changes that are occurring in the brain from basically conception until young adulthood. And one of the things that I wanted to notice, or want you to notice and to point out, is that there are regional changes in the brain. And I'll show you pictures in a minute to make this a bit more concrete, but prefrontal regions of the brain are being fine-tuned at a later or slower time frame than very basic regions of the brain that allow us to see and to move. So the prefrontal cortex, which I'll come back to time and time again, is very important with regard to our ability to engage in rational thought, and it's very important in self-control or helping us to regulate our emotions and our behaviors. So all of these changes are taking place at the same time that the brain's being marinated in gonadal hormones. And also we're seeing a number of changes that are occurring in neurochemicals, in blood flow, and in this white matter or fatty substance that's referred to as myelination that allows regions of the brain to communicate more efficiently with one another. Now, with the advent of human brain imaging, we're now able to, if you will, look under the hood of the brain in action, and almost in a voyeuristically way with regard to looking at the teenage brain in action. So we can take pictures of them when they're in an MRI, and then we can look at what areas in those pictures of their brain are more active than others as they perform games or simple tasks that we ask them to perform. We spend a lot of time making the imaging environment a child-friendly place. It's actually the case that young children are less claustrophobic than adolescents or young adults. They like sort of climbing inside what they feel like is almost a spaceship. We spend a bit more time trying to acclimate our teenagers. This is just showing you a picture of a child and a 60-centimeter board, this is about the size of the opening of most MRIs that we're using for research around the world. Many groups, including our own, use pre-simulators to make the experience as child-friendly as possible. And you can get these collapsible play tunnels that are about 50 to 60 centimeters in diameter. And for young children, you can let them actually run through this and get an idea of how big it is. You can play the sounds the scanner makes. And really, we're making this not anything like a clinical MRI, if any of you have had one. I know I have. Basically, our motto with imaging studies with children is you do everything but feed them popcorn. And this is just showing you a child in a pretend scanner. And again, you can see how much room that they have. But when they perform tasks, they actually end up looking up into a mirror so they can see a screen that we place here. Let me just show you that. So here's a screen we can put above the individual for them to play. They can press a button to answer questions we have or to play the game. And you can Velcro that on their hand so they don't lose it or have to look down at it. And then this is the individual lying comfortably in the magnet as we take these pictures. After these procedures for making the child and also the adolescent, we take them through the simulation process, not through the play tunnel, but through the pretend scanner. And we allow them to get comfortable, make sure that they're okay with the procedure, and then we can take real pictures and get movies such as this in terms of just looking at structure. These are images from the National Institute of Mental Health, where I used to be a postdoc. I want to just play that movie for you one more time. Basically, the areas that are turning blue are developing before those that are taking longer to turn blue. This measure is really looking at the cortical surface, the outside of the brain, how thick that cortical surface is. And it's a little bit easier to follow if you look at these images, as if you're looking down on top of the brain from roughly 5 years of age to the early 20s. And notice that this motor cortex area and this visual cortex area, those are regions that allow us to move and see, are developing earlier than areas in the prefrontal cortex, see, that are continuing to change in a very slow way up until the early 20s. So it makes logical sense for those areas that allow us to move in our world and to see it, for them to develop before we use those same reasons to guide our future behaviors and also to regulate them. Again, this prefrontal area, as I mentioned before, is very important in regulating our behavior and rational thought and thinking about the future consequences of our behavior too. My colleague Jay Geed has talked about this region as being a time machine. The prefrontal cortex helps to link what we've learned in the past to what we're going to do or plan to do in the future. But it's not just about the outside of the brain, particularly when we think about changes that occur from childhood into adolescence. During that time period, we're seeing a lot of changes in emotional reactivity. And what we have discovered over the years is in this glass picture of a brain, or picture of a glass brain, we see where there's fuchsia color that there are significant changes in the prefrontal cortex, but there are also subtle changes that are going on in these emotional centers that are very important in desire and rage and fear. And it's really these functions that we think of in terms of adolescence, and often it is these specific functions, particularly being able to process fear and threat and recognize when a threat is no longer present, that appear to be disrupted in so many of the anxiety and stress-related disorders that this talk today is most relevant for. This is just illustrating for you in a movie, in a glass brain, the deep red structures where you're getting dynamic changes that are occurring during adolescence before the prefrontal cortex in green here is able to fully mature and project to that region to help quiet it when appropriate. So basically we describe this transition from childhood to adulthood as almost a tension or imbalance between these emotional centers of the brain and those that regulate it. And again, they're on a different developmental trajectory, and that's in part the imbalance. But there are a number of factors that can exaggerate or exacerbate that imbalance. And these include both environmental and genetic, but in the latter part of the talk, I'll be focusing specifically on some of the environmental factors. Now, how do we actually measure brain activity? How do we try to begin to measure how reactive a person is to emotional information and how well they can regulate their emotions? Well, one way is to show these cues of threat. I don't think anyone looking at this image is afraid of this picture. But what we've learned over a lifetime of experiences with phases such as these is some form of uncertainty about the threat. Somewhere, something is usually about to happen, or something bad is about to happen when we see one of our peers or a child or someone express this fear. And even without our conscious awareness, it's cues such as these that actually alter our behavior, even without us being able to articulate that. And let me just show you that from some laboratory data. In this next slide, all I'm showing you is how quickly we typically respond in a laboratory setting if I just asked you to press a button whenever you saw a fearful face versus a neutral face versus a happy face. So the higher the bar, the longer it takes the individual to simply press a button. And you see that they're taking much longer to press a button when they see this face as opposed to either of the others, and they're about equal. Now, after the experiment, when you ask an individual, did you feel like any of the faces were making you do anything different? Were you faster or slower? None of your subjects can tell you. It's just a subtle difference, but it's definitely occurring. So this is how we're processing information differently. We see it. We're trying to interpret what is the emotional significance for us in this environment. In the laboratory, nothing bad's going to happen to these individuals, so they should get quicker and quicker the more that they see these faces and sort of say, all right already, nothing's going to happen to me now, this doesn't mean anything, and so that emotional response should quiet. And the way that we measure that is, again, to take pictures while they're performing tasks such as these. And there are two areas of the brain that are part of a brain circuit. The amygdala, that's very important in detecting the emotional significance of cues. And then a portion of the prefrontal cortex that I was talking about before that helps to regulate actions and emotions and behavior. So the very area that projects to this emotional center is doing something quite different than what the amygdala is when detecting simple emotional stimuli. And they're both correlated with how quickly you would detect one of those fearful faces. But let me show you how different that pattern is. In this emotional center, if you're an individual, each one of these are individual data points, and this is how fast the individual is, and this is how much brain activity there is. So if you're an individual that's activating this region a whole lot, you're a lot slower to detect that fearful face relative to a happy, smiling, or neutral face. So this is in contrast to when you activate the prefrontal region. So again, reaction time is here, and the amount of activity is here. If you're an individual who is activating this region a lot, so see that the activity is very high here. The more you activate that region, the faster you are. So these are slower responses, and these are faster responses, suggesting more regulation of that heightened emotional response. And let me try to unpack that for you just a little bit more in terms of how this pattern of activity changes with development. So first, let's just focus on this emotional center of the brain that's activated when you see a cue of potential threat. This is the amount of activity. And look, these are children and these are adults. You don't see any of these individuals activating as much as we see as a group the adolescents are. So they're activating more to the potential cues of threat. And this heightened emotional reactivity probably wouldn't surprise most individuals and most of you who have gone through adolescence. We have a bit more emotional reaction to things or anyone who is experiencing their adolescent, particularly early adolescent ages now in their family. But what's very important for the adolescent to learn to do to help the wiring of the brain so that they can learn to regulate their own emotions when they become more independent from their parents is for them to bring the signal down. So what I've plotted for you here is the beginning of an experiment. It's the exact same plot, and then at the end of the experiment, within 20 minutes of when they started. And we see that there is much more activity. It's higher for all individuals. But then over time, with repeated exposures, it's almost as if the brain says, all right already, nothing's going to happen here, and it begins to quiet down. And it's this ability to regulate that emotional response that is associated with anxiety symptoms. So if you look at individuals, none of whom have a diagnosis of anxiety, but who say in everyday life they're experiencing a lot of anxiety, we actually see the degree to which they can quiet it. This is a simple difference score, percent difference score of early versus late trials. The higher it is, the more it means the emotions, the emotional reactivity decreased. The higher that is, the less they rate themselves as having high symptoms of anxiety. But what I really want you to do is to focus on this quadrant of the graph, and that is a sample of individuals who rate themselves as having a lot of anxiety each day. And what you should notice here is these are negative scores. If you have a negative score, what that means is not that they quieted down, not that there was a decrease from early to late trials with all of that repeated exposure and trying to desensitize them to the fearful information. What you see is they actually sensitized. So if it's a negative score, it means that they actually increased activity in this region over the course of the experiment as opposed to showing a decrease. And now this is in individuals who don't have a diagnosis, but these data are very relevant for children who do, and I'll come back to that in a minute. But what I first have to unpack for you is what is the part of the circuitry that helps regulate the activity in this region of the amygdala. It's the prefrontal cortex, right? And that's exactly what we see when we do an analysis that tells us how well areas are activating together or are connected. We see that the better able you are to quiet or habituate that amygdala that's so important in emotional responding, the better you're able to do that, the more inverse coupling between the prefrontal cortex and the amygdala. That means essentially that the more the prefrontal cortex is active, the more quiet the amygdala should be. And if you don't have that pattern, if you have more of a positive pattern, it's harder for you to regulate your emotions. It means they're both activated and one's not coming down. Now, if we look at individuals with anxiety, we see the same. This is as if you're facing the brain, looking right into it. This is this emotional center again. We see that more activity in this region in children and adolescents who suffer from anxiety relative to healthy controls or who have a diagnosis of anxiety relative to those who do not. And what's important whenever you see these patterns of activity is how that really does relate to the severity of symptoms. And that's plotted for you in this next graph. Basically, this is the child reporting everyday ratings of anxiety. We refer to this as a scared score using that particular survey. And this is the amount of activity in that amygdala. So you see there's a positive association here. The more anxiety, the more you're activating that region, and the less able you are to bring that activity down. So I've shown you a lot of empirical data as a basic scientist myself, but what I want to do is show you a movie that might unpack or summarize for you the information that I have presented so far. And so in this movie, I'm basically going to be showing you, this is as if you're facing the brain, and you're going to look at the pattern of activity, and I want you to focus again on this area deep in the brain, these emotional centers, and how it responds each time I present one of those fearful faces. So you'll see the presentation of fearful faces over here, and then you'll see with repeated exposures how someone who doesn't rate themselves as being very anxious responds to that over time. So here what you're seeing is activity. It's bilateral activity. You have two hemispheres of the brain, and we're seeing in both amygdalae. Each time you present that cue of potential threat. And then as it turned blue, that is the pattern of activity returning to baseline. So you see initial activation as you're trying to understand, even if you're not consciously aware, the brain's trying to understand the emotional significance of this cue. And then with repeated exposures, it goes to baseline. Now, that's in stark contrast to what we see in individuals who support themselves as having high anxiety throughout the day. So initially, it looks very similar to what we saw in the individual before, but what you'll notice is even with repeated presentations of this, we're not seeing that activity decrease. It's that they can't quiet that emotional reactivity. They remain reactive, and it's more difficult for them to regulate that emotion. So it's data like these that actually have suggested to us, sort of as an interim summary, is a hallmark of emotion regulation is the ability to us to learn when cues or certain contexts no longer signal any potential threat. And this is the very basis of what exposure-based CBT, cognitive behavioral therapy, is based on. The principles are the individual comes into the office, the clinician helps to determine what are the triggers, what's causing the anxiety, and when they can really target on a few triggers or what's causing the anxiety, then in that safe environment of the clinician's office, they can work with the individual to try to desensitize them to those triggers. So basically desensitizing them by exposing them repeatedly and allowing them to learn how to better cope in managing their emotions to those cues. But the differences that you saw in the movie that I presented before between an individual that has low anxiety and those who say they really are experiencing high anxiety, those individual differences really beg for us to begin to have a better understanding of what might make an individual more anxious during this time. And there are a number of pathways to anxiety and stress-related disorders. There are a number of genetic and environmental factors that come into play. But again, today we're just going to focus on some of the environmental factors in the interest of time so we can open it up also for some questions. The particular stressful early stress environment that I'll be speaking to today is that of being reared in an orphanage or the experience of institutionalized life. And basically, this is an experience that involves fragmented caregiving, and typically from very few caregivers relative to a number of children. And there also a high turnover often in these orphanages and institutions of many different caregivers being available, but only a very small number at one time for a large number of children. We had an opportunity to examine the effects of this type of early life history, which is unfortunate but is occurring, to try to learn about how fragmented early caregiving could lead to changes in emotion regulation. And so the children that came into this study had been in the institution for 6 to 60 months, the oldest was 5 years of age when they were adopted out of the orphanage. And this work was done here in New York City. And many of these children were adopted to families from orphanages in China and Russia and other countries. But we would only test them after they'd been in the United States for at least 2 years, because for some of these children, almost everything they experienced was novel, including even just texture in their food. And so providing them time to acclimate to this new culture and also to adjust to their new family was very important. And I must at this point thank these families for participating in the research I'm going to describe, because already the parents who are adopting so many of these children are super parents and really giving them opportunities that are tremendous and that they've not experienced before. The type of behavior that we wanted to measure to get a read on emotion regulation was, again, using these cues of potential threat to see how they process them. But if you want to look at emotion regulation, one way to do that is to say, how well can they ignore or suppress them? And so before I showed you with repeated presentation over time, they tend to suppress their emotion toward information such as this that suggests threat. But in this population of individuals, we are very interested in how well they could simply ignore. So we said, this is a non-target, don't do anything at all, and the rest of the faces you want to press to. And it was our hypothesis that they would have less ability to really regulate their emotions and behaviors because of the really the lack of contingent feedback and caregiving that they'd experienced early in life. And so we thought that what these cues might be more of an experience for them. Even when they were ignoring them, they would see them as quite salient. And so when you have a task like this and you're asking them to present or press a button when they're presented, what you see is that children who were adopted from these orphanages abroad were showing much slower reaction times to detect these cues when they knew a rare threat target was going to happen, as if in anticipation they were being slower to the cues. You didn't see the slowing in response if they were anticipating a happy face. It was only in the context of them anticipating negative information. And we didn't see that in the non-adopted children who had grown up within typical families. Now, the next thing that I want to show you is how this behavioral pattern is actually represented in the brain, again, using the technique of functional magnetic resonance imaging that I described before. And basically, what we see is that what's in blue are areas that are being recruited more in non-adopted children, and this is an area in the prefrontal cortex that actually drives your ability to eye movements and actions to suppress those. But the area that's in red is the area that was more activated in the previously institutionalized children. So you're seeing a different pattern of response here. And also I should just note that we're seeing it again in both hemispheres in this amygdala emotional center that I've described before that's so important in terms of evaluating the emotional significance of stimuli. In this task, they should be ignoring this threat cue, but their readout, the neural signature, looks as if they just can't suppress that relative to non-adopted, healthy volunteers who were the same age. But it's not sufficient, really, for us just to show differences in the brain. When we see differences in the brain that are actually linked to severity of anxiety symptoms or linked to behavior that's so critical for social interaction and emotion regulation, that's when we feel like we're informing the, really informing the field and telling us a bit more about changes in the brain that are leading to social and emotional delayed development. And so what I'm highlighting for you now is a complicated slide. And in fact, because it's so complicated, I'm going to back up a second. I'm going to tell you two other measures that we were simultaneously obtaining from these children. The first is when the children came into the laboratory, into the institute here, first we would go through all the games and tasks and things that they were going to do, and we explained that and made sure everybody knew what they were going to do and got their consent. And then they would go and play with one of the research staff for about 10 or 15 minutes, and they'd engage in a puzzle game or any games that we were actually using to measure their cognitive and emotional development. Usually we made the first one fun just to get them acquainted with the environment. Then they would come back and we would say, why don't you just check in with your mom or your dad, whoever brought them in. And during that time, we actually videotaped their interaction with the family's permission. And so we wanted to see when they'd been separated from their mother or father, how adopted mother or father, how much interaction they had with them when they came back and were with them. And at this point, remember, the children were adopted anywhere between the first year and 5 years of being institutionalized, and we made sure that they were all in the United States for at least 2 years. So we're really talking about children in school up to about 15 years of age, so children and adolescents. So that's one measure. How much eye contact are you having with your adopted parent if you were previously adopted from an orphanage, or with a parent if you actually were never adopted? So that was our comparison group. And then the other measure was we had a different task where we would just show them pictures that were either smiling faces or fearful faces or kind of neutral, bored-looking faces, and we would see how much time they spent looking or having eye contact or gazing at the eyes as opposed to other parts of that picture. And we could measure that carefully with eye tracking. So that's what we saw was associated with the amount of activity in this region for the children who had been previously institutionalized. And that's what I'm showing you here. So on this side is how much live interaction or eye contact they had with an adopted parent. Now we're just looking at the activity here in the amygdala. If it's a whole lot, which is on this end, the more activity, the less you see they had eye contact with the parent. And also on this side is the eye tracking. So again, you see the less they spent looking at the eyes of pictures, the more activity they also showed in these regions. And so this suggests this ability to quiet emotions that we can get a readout in the brain are very important in terms of the observed behavior that we're seeing with people, and also in processing social cues that are so important for development and our ability to engage in social communication, but also so important particularly during the adolescent years when they're having to negotiate social interactions without their parents as much and having to read those cues. However, it's the case that children who have been adopted from orphanages abroad, we know so little about them. And many of these studies, it could have been the case that there were environmental or genetic factors that we were unaware of besides just the fragmented care that you typically get in an orphanage. In other words, they could have had a genetic loading for disorders, or they could have been exposed to drugs or alcohol or toxins that might have led to this. So we've begun to do a number of parallel studies in mice that kind of mimic some of the situations with humans. Never are you going to say that a mouse is equivalent to a human, but we can at least get a bit more control over these experiments. And I wanted to highlight for you how we've done this in a mouse study looking at stress. Basically, the stress manipulation, and this is a video that's sped up, was to take away the nesting in the dam's cage. And you might be able to see, if you've got fair resolution, that there are little mouse pups underneath the dam here in the control situation, and there's plenty of nesting in this cage. Here you see less of that fluffy nesting, and the dam is running around as if she's trying to collect that nesting for her pups. But her pups are here and there, and she's spending much less time licking and grooming them, or really spending less unfragmented time with them. And we actually show that here in this graph. So this is the amount of time that the dam or the mother is spending on the little mice. And you see in the mother that's not stressed, she's spending significantly, almost double the time that the stressed mother is. So this is very important in terms of thinking about how environments can lead to stressful behavior that can then subsequently lead to less capacity to be able to carefully care for offspring. Now, in addition to trying to come up with a situation or stressful event that would parallel what we were seeing in the orphanage, that is the fragmented care that you see in the orphanage, we also wanted to try to come up with some way, because we couldn't tell the mice what we wanted them to do, but we wanted to test the offspring to see how they were impacted by their mother's stress, and they're spending less time grooming and nurturing them. So how could we get them to engage in a task when they knew that there was possible threat? And one way to do that is to try to find something that mice really, really love, and it turns out they really like condensed milk, condensed sweetened milk that we put in a lot of desserts here. And so basically the task that we used was one in which we put a nozzle in their home cage and trained them where they could get the condensed milk. And we did that for 3 days of training. And then on the 4th day, we measured how quickly the mice actually ran to the nozzle to get the condensed milk. And we compared that to when we placed them in a novel cage that was well lit, which mice don't usually like. It usually, it's novel and it's well lit, so it might be potential threat. And we measured how quickly the mouse would respond to the nozzle in that situation. And before I present the results, I want to remind you again of what we saw in our children who had been adopted from orphanages before. And that is, remember when they were in the situation where they were responding, but they knew at any moment this rare, fearful face might pop out, they were much slower than those children who didn't experience this early stress. And we saw that that was related to the amount of activity in this emotional center of the brain that failed to quiet with repeated exposures. In mice, we saw the same thing. They were mice that were stressed, where we'd taken nesting from the dam in the early life during the first 21 days of life. That's basically, they're weaned about 21 days, and we wanted to mimic the timing to be similar to when the children were adopted from the orphanages with childhood, and that's when we ended the stress for them. And we see they show this slower tendency to go to the condensed milk that they love, but they're slower to go after it relative to those mice who weren't stressed. And when we look at CFOS, we can also measure amygdala activity. I don't know if your screen will have enough resolution, but there's a clear difference in the amount of amygdala activity in the stressed animals relative to the non-stressed. And these are the offspring, again, experiencing the stress of the dam or the mother of the mice. It was also the case that with mice, they develop quickly. So we could look at their behavior and look at different groups of mice to see how this changed from the early life to adult life. And we saw this heightened activity in the amygdala was even present in the adult. And remember how the prefrontal cortex is supposed to be so important in regulating these emotions? We even saw that in the adult, the prefrontal cortex activity actually increased, but this was not sufficient to actually suppress this emotional response. So again, the stressed animals are continuing to show more of this emotional reactivity, and even with maturity, they're finding it harder. And when that prefrontal cortex is fully developed, they're still finding it harder to suppress or habituate to that potential threat. most recently, collaborators of ours, but in an independent study, Nim Tottenham and Dylan G. at UCLA, have begun to examine the connections between the prefrontal areas and this emotional center, the amygdala in the brain. And what they're showing is that it looks like there's a sensitive period of neural development that underlies emotion regulation, and that early stress may actually lead to a closing of that sensitive period. So these are healthy individuals, and how, remember at the beginning I told you there was a transition into a inverse relationship or inverse connections between the prefrontal cortex and the emotional center of the brain. And the normal tradition is to see that right around adolescence. But we're actually seeing that earlier in children who have been adopted from these orphanages or experienced early life stress. So presently, what we're trying to do is determine ways in which we can try to find ways to open these windows or to work to really change these fear memories and emotional reactivity in different ways. But let me just kind of conclude by saying, in the animal studies, we're seeing these early and lasting alterations in amygdala circuitry and function with prolonged stress. It's this circuitry that's implicated in so many of the psychiatric disorders across the board. However, here I'm showing you ones that are specific to anxiety and stress, and showing you an example of how a stressor, even when it's removed, and even after development of the prefrontal cortex, we're still seeing those effects. So the charge to scientists and part of the foundation in the work that we fund is to think of novel ways in which we can detect individuals at risk early before these sensitive periods in development really close in the brain. That's very important to intervene early. And to also begin to work on novel treatments which were not there, but were just almost at the surface of novel treatments emerging to help children desensitize themselves and to help regulate their behaviors in a way we've never been able to do before. And what I want to do now is just end by acknowledging so many different partners and much of the research that I presented today, and to also highlight a potential conflict, although I didn't talk about any work that I do on MacArthur Foundation of Law and Neuroscience on the adolescent brain and culpability. I do want to acknowledge I get research funding, and I'm a consultant for that group. And one more time, I just want to acknowledge the incredible work that's driving new interventions and treatments that I didn't get to today and other scientists are working on that the foundation has funded that are so important. Thank you.
Thank you, Dr. Casey, for an outstanding presentation. You were able to take very complicated work that you and others have done and present it in a way that non-scientists can have an understanding. So thank you for everything that you've done and for today's presentation. You sort of ended discussing the potential for some novel treatments, and we've had a number of questions about what types of treatments may be coming down the road that can help families with kids with this kind of experience. So I wonder if there's a little bit that you could say to us to, even though it's not ready for prime time yet, what types of novel treatments are down the road?
So I'm going to stick with some of the behavioral treatments, but, because much of the work I talked about was for children and adolescents, and any time we can be successful with behavioral treatments as opposed to medications, we want to go that route. Unfortunately, behavioral treatments don't always work, and so there have been a number of novel drugs that are beginning to come on the market. I'm not going to speak to those, but cognitive behavioral therapy that I mentioned before, where you are trying to desensitize the child or the adolescent, and even an adult, you're trying to desensitize them to cues in the environment or particular environments that are causing them undue stress and anxiety. they have in part been successful, but in part it's been difficult because some age groups and also some individuals with certain genetic variants don't have as much of a capacity to desensitize. So I showed in the movie how an individual with low anxiety was able to eventually kind of get over having an emotional response with repeated exposures to a simple threat cue. That's what you would do with therapy, only desensitizing them to, say, separation from a parent, or if they had a particular social phobia. But we are beginning to see in the research that adolescents and also individuals with certain genetic variants are not very good at being able to extinguish these fear memories. And so what we need to do is to find ways where we can actually bypass the requirement of the prefrontal cortex to help them regulate those emotions and extinguish those fears. And one of the ways we've begun to do that is to build on recent basic science that now shows that our memories are not static. They can change with time. In fact, every single time we recall something, it becomes fragile or labile. And there is a window of time when you recall a memory when you can alter that memory and attach new information to it. And so what we and others are beginning to do is to try to use the timing of that special window when you can change a memory at the very site of the memory, which would be at that deep emotional center structure of the amygdala, and never need the prefrontal cortex to try to help regulate it. This is all in the timing. We know that critical window is about 10 minutes after you activate the memory, and it lasts for about an hour. And so ways in which we're trying to get clinicians to test this as a proof of principle is the only change you're making is the timing of when you start desensitizing them to their fears or phobias. So the patient comes into the office, the clinician reminds the patient of why they're there. That reactivates the memory, right? Then they're waiting to get into that sweet spot where you can actually reconsolidate the memory and actually add new information to it. So they wait for about 10 minutes, but they're not doing anything. Usually what clinicians are doing is they're trying to make the patient feel safe and talk about anything positive that might be going on in their life that you know, in previous sessions they've learned about. Then when they get into that sweet spot in time, about 10 to 15 minutes after they've reminded them of the fear and why they're there, then they can start using desensitization to potentially attenuate or erase that fear memory if possible. And we think that this is going to be more effective based on some of the basic research that's come out of Liz Phelps and Joe Ledoux's labs at NYU, and also our own working with adolescents here at the Sackler Institute at Weill Cornell. I know that was a lot of basic science, but I hope that was a little bit helpful in terms of how we're trying to move basic science more quickly to the clinic.
I think it is, and it's a great example of how very basic science information can be used to help clinicians do a better job in their treatment, that there are physical effects that are in the brain that basic science can show us that could then lead us to do a better job as clinicians in terms of the treatment. And we're not talking about medicine here, we're talking about approaches to talk therapy and ways to intervene to help people. So I think it was very important for you to share that level of detail.
And I also think this is very important with regard to the foundation and really us all being one family with bringing together of clinicians and scientists and the families together in driving this research.
Absolutely, absolutely. I'm going to just ask one more question given the remaining time, and that is, even at this point in time, and I'm not asking you a clinical question as a clinician, but just guidance to parents. If the parent is concerned about their child, whether they be one of the super parents that you refer to who are adopting these children, or they have a child that they gave birth to that they have concerns about, what types of things should they do for their child?
So I think it's important to, it's always important to get professional advice when parents are troubled about their child's behavior, but it's also important for us to recognize that during this transition from childhood to adolescence, when you're seeing significant changes in hormones and you're also, the child's having to deal with physical changes and how people are treating them differently, that can really change their emotional capacity, and to recognize that they do need opportunities to regulate their emotions. It's not always the case that children are capable of doing this even when given the opportunity. My 11-year-old son, when he first came home from school, right as he was emerging and going into puberty, he came in and used words that even I don't have not used, and it just shocked me so much I couldn't say anything because these were bad words that we don't routinely use in the house. And rather than say anything, he just looked at me and he said, basically, oh, right then, you know, it was like, you're not even going to respond to me, as if he was upset with me, but I couldn't respond because I was in shock. He went upstairs, and for the next hour he was in his room, but it was also at the same time that I was making dinner. And when you're transitioning into adolescence, you need a lot of calorie intake, too, through these growth spurts. And he came downstairs, and he didn't say anything at all, except he just nuzzled next to me. And I just said, rough day. And he didn't say anything, but he did proceed to get a plate of food and sit down, and then it was a nice dinner. So parents are so stressed these days. Often when our child does something or says something we don't expect, we tend to just fly off. And it's important for us to take a deep breath and for them to learn how to take a deep breath so that they can begin to learn to regulate their behavior. But there's so many good strategies for trying to get children to cool off, to take that second to take a deep breath, and also trying to help children get engaged in activities that allow them, it's not just keeping them busy, but sports, meditation, things that allow them to regulate their mood, their behavior, the way that they're thinking, are very important too. But then when these behaviors continue, it's very important to try to seek advice. So early on, we can provide coping skills to make it somewhat easier for children, or ways in which we can help them. Because helping them early is so important, and not treating can lead, again, as we said at the beginning of the talk, really to chronic symptoms down the road.
Well, I just want to say, both as a psychiatrist and as a parent, the guidance that you just gave is beyond right on target, and it shows why you're not only such a great researcher, but obviously a great parent as well. And I appreciate you sharing both the personal and the research and professional so that people can use those suggestions in their own life and help their own kids. So thank you for doing that.
Thank you, Jeff.
Yeah. So I just want to, again, say thank you for this extraordinary presentation, and thank you for the work that you've been doing. I also want to thank our audience for joining us today. The Foundation, through its research grants, is dedicated to improving the lives of people with a wide variety of psychiatric conditions. All of the research we fund is made possible through private donations. So if you'd like to make a gift, please visit the website bbrfoundation.org or call 800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the webinar page on our website. I hope you'll join us again next month when we'll hear a presentation by Dr. Andrew Nierenberg, who is a professor of psychiatry at Harvard Medical School and co-director of the Bipolar Clinic and Research Program and associate director of the Depression Clinic and Research Program at Massachusetts General Hospital. He will present a webinar entitled, "Is Bipolar Disorder an Energy Disorder? Evidence and Novel Treatments?" That will take place on Tuesday, October 14th at 2 p.m. Eastern Time. And for anybody who's in the Washington, D.C. area, on September 16th, we invite you to join us for the Foundation's upcoming conference, Discovery to Recovery, A Path to Healthy Minds, featuring keynote presentations by Congressman Tim Murphy, as well as a presentation by former NFL player and Super Bowl champion Keith O'Neill, who shares his experience living with bipolar disorder, as well as a number of extraordinary scientists speaking about child and adolescent anxiety, bipolar disorder, depression, PTSD, and schizophrenia. For more information about this event, please visit our website as well. I want to again thank you all for joining us and wish you a pleasant rest of the day. Take care, everybody.